Jamala, the floor is yours.
Okay, thank you very much, Mr. Shaim Sharkar, Dr. Shohail Abdullah, for inviting me for this interesting webinar from Jahangirnagar University. And I'm excited, when I heard that there was 650 and more registration requests, and you could not also go on registering people because of your limitation. So it is really exciting for me to be here and to present some of the exciting fields of genome editing in front of this much of audience. Not going into detailed introduction. So you know all about my title of today's talk, the basics of genome editing, what, why, and why, and how. So I would basically focus on the basics of genome editing and also, I would briefly mention about different kinds of advanced techniques and some applications also. So this is my outline of my presentation. So in part one, I would mainly focus on basics, and this is the main motto of this presentation, to bring the whole genome editing arena into your knowledge in a very simplified way. So in these basics, I would cover what is genome editing and why is it necessary. And then I'll go for explaining briefly what are the different editing platforms that we could use for genome editing, including meganuclease, then ZFN, and then TALEN, and obviously the most exciting CRISPR-Cas9 technology, by explaining how they have been originated or how they have been discovered from a bacterial system and they have been repurposed for genome editing. And then I would highlight a few limitations of this technology, obviously. And then in part two, I would now go beyond the normal genome editing knockout system. So I would explain some of, in few slides, that what else we can do with the CRISPR-Cas system. And then in part three, I would give you a glimpse of my work with precise editing and what we are doing now. And in part four, I would be giving you some outstanding examples from different fields, starting from basic biology, and then agriculture, and then obviously therapeutics, human therapeutics. So this is the screen, and in these two corners, you can see the two most devastating and dangerous names here. In the left, it is the coronavirus or COVID-19. In the right, it is the AIDS disease or caused by the HIV virus. You know, these two viruses are common in many sense, because if you see here how the coronavirus infect very easily to human cell. So they use our own protein. You know, our human cell membrane has a protein. That protein is called ACE2 protein. That is a receptor protein. And what this virus do, they come and bind to this receptor protein, and they use this protein to enter into our own cell. So they use human protein to enter human cell and then destroy the human cell, infect the human organs, and then ultimately it could be lethal. So similarly, HIV virus also insert, they enter into the white blood cells by being attached by a receptor protein that is called CCR5, and other co-receptors are also there. So in case of HIV also, they also use our own protein receptor, that is CCR5 here. So this, both the virus, they use their own protein. So you could easily hypothesize one thing here, the how to inhibit these two viruses by altering our own system. So suppose if you have some mechanism to specifically reduce the number of those protein in our human cells, or maybe just eliminating those protein, but is there any specific mechanism? So the question is there a way to disrupt specifically those receptor proteins that are utilized by those viruses to infect the human cell? So if we could just disrupt those kind of protein and reduce their level, we could probably able to inhibit those viruses. So that's the simple, but the question is there a way? So I would be discussing my whole presentation about that way, and that way is obviously genome editing, and I would be talking in details in my presentation. So in all my presentation, I always start my presentation with these introductory slide to rejuvenate ourselves about the concept of cells, DNA, chromosome, and all these things, and what is gene and DNA. So human body is nothing but around 37 trillion cells, and like the higher eukaryotic plants like rice plant here, also made up of trillion of cells. So here is an example of a plant cell. And this cell has a nucleus. All of you know that nucleus is the head, is called as the head of the cell. Why it is called as the head of the cell? Because it performs most important function of the cell, because it contains chromosome, which is known as our genetic material, because this chromosome is made up of long thread of DNA, and this DNA is coiled around a special protein that's called nucleosome protein. So chromosome is nothing but long thread of DNA and coiled around that protein histone. So simply, to make it simpler, just think about chromosome and then DNA. But the long thread of DNA, all portion of DNA are not so important according to the present knowledge. So some portion of that DNA is functional, or it is important for its function, and this portion or unit of that DNA. So the functional unit of DNA is called gene. So here it is green highlighted. This portion is the functional unit of that long DNA, and it is called a gene. Because this gene, when it expresses, it produces a protein, and this protein governs a character, so directly or indirectly. So this gene is expressing a protein, and that protein governs the character. So one gene, one protein, and then one character. So now, so what is in that DNA? Our DNA is nothing but the different combination of the sequence of 4 different kind of letters. One is A, G, C, and D. That is adenine, guanine, cytosine, and thymine. On that makes the genetic code, and this genetic code controls almost everything in a cell's characteristic features. So if you go then, now the question is, as I explained, that the protein is produced from a specific gene. So I told you that coronavirus and then the HIV virus, both of them uses our own receptor protein. So the researchers have already deduced what are the gene responsible for producing that receptor protein. So if we could specifically lock that portion of the DNA or lock that gene, so we could be able to reduce the level of that protein, receptor protein. So if that receptor protein is no longer available or in a very less available mode, then the virus would no longer be able to utilize that protein to infect our own cell. So then our aim is to specifically block that portion of DNA that produces those proteins. So okay, is there a way to specifically block a targeted portion of gene? The way obviously is genome editing. Now we have the technology to perform that kind of a specific function. So if I talk about genome editing, there are 4 well-known platforms to genome edit. The earliest one is meganuclease, and then comes ZFN, and then comes TALEN, and this is the most popular one, that is CRISPR-Cas9. So if you see, the earlier 3, that is meganuclease, ZFN, and TALEN, all 3, they depend on protein and DNA interaction. So if you want to edit the DNA or the gene, then you have to have a protein-DNA interaction. But in case of CRISPR-Cas9, it depends mainly on RNA-DNA interaction by simple Watson-Crick base pairing. So here there is no protein actually specifies the targeting of that genome editing technology. So that's why CRISPR-Cas9 is a little bit easier to use, and that's why it became very much popular. So I was lucky enough to meet this is Daniel Poitras, who was the pioneer in TALEN technology. Daniel Poitras, I was giving a lecture in the University of Minnesota, and this is another with Jennifer Doudna. Jennifer Doudna is the pioneer of CRISPR-Cas9 technology. This is in Cold Spring Harbor Laboratory in New York. So going to details into the meganucleus, the first one. You know, the meganucleus is nothing but a simple restriction endonuclease that we use to cut our DNA in case of cloning experiment, you know, HindD3 and then BAMH1, then EcoR1, all of these things. So all of you students probably know that. But meganucleus is why it is called meganucleus, because it has a long recognition site. Normal restriction enzymes like HindD3, BAMH1, they're hexacutter. So they recognize 6 specific nucleotide or 6 specific base pairs in the DNA, and then they go there and then they make a cut. But meganucleus, they recognition side up to 40 base pair recognition. So if you see, calculate in the genome, even in human genome, some meganucleuses have only one side, and in other genome only 1 or 2 sides. So it is highly specific. But the problem is, you have if you want to make a cut in the DNA. So let me remind you one thing. For the first step of genome editing is to make a specific double strand break in the DNA where you want to edit your DNA. So up to this, just keep in mind that double strand break is necessary for genome editing. But you would be understanding in my later slide why double strand break is necessary for genome editing. So now meganucleus, what I chose sequence is 40 base pair long up to. So it has very specific nature, but it is very difficult to target every gene and whatever gene you want to target to make a double strand break. But you have to engineer this meganucleus to change the recognition sequence. So suppose you have a meganucleus that has a recognition site in gene A, but you want to use that meganucleus for editing gene B or for making a cut in gene B, but you cannot use that same meganucleus because it does not have that recognition site. Okay, so now you have to engineer that protein to change the recognition sequence, but it's very difficult because you have to engineer the whole protein to change the recognition site. So that's why it has a limited use. Now coming to the second, that is zinc finger nucleus. You know, zinc finger is a protein motif and which is normally present in many transcription factor, and we know that transcription factor protein, they come and bind to a specific DNA. So this zinc finger motif has specificity to bind with DNA. If you see here, one zinc finger can bind with 3 base pair of DNA. So here for 3G, there is one zinc finger, another zinc finger, they can bind another 3 base, and then another zinc finger have bind another 3. So if you use 3 zinc finger, then you could just target that zinc finger to bind with 9 base pair. So now, okay, this zinc finger can bind with a specific DNA. We know we can engineer that kind of protein, but how it would make a cut in the DNA? It does not have cutting ability. So that's why if you see, this zinc finger motif is fused with another endonuclease enzyme that is called FOC1. So this FOC1 fused with zinc finger, this is right ZFP and this is left ZFP. Now you have question why we should use both ZFP, why not only one single ZFP? Because this FOC1 is very specific in nature, and this FOC1 cannot make a cut until and unless it forms a dimer. So in order to form a specific dimer, so you have to use 2 zinc finger in from the 2 opposite directions, so the FOC1 come together and then they make a cut. So that is why that is how ZF makes a cut to your specific gene. So you have to design your own ZF, that is zinc finger motif, to target your gene of interest, because you know the sequence, and for what sequence, what kind of protein, you have to design that. So here also protein engineering is needed. Now coming to the talent, next, the third portion. Transcription activator like effector nucleus. So this TALE is the effector, TALE, transcription activator like effector from a bacteria called Xanthomonas oryzae. These bacteria infect rice plant and causes a disease that called bacterial blight. And these bacteria secrete this protein, and this protein goes to the rice DNA and it binds specifically to a promoter, and it increases the expression of certain genes. So it acts as like transcription activator, but it is secreted from the bacteria. Okay, so the main point that I want to highlight here, that this effector protein also has specificity to bind certain DNA, not to all DNA. Okay, and that has been utilized by the genome engineer, intelligent scientists, they have used it for genome editing purpose. How? They fuse the tail repeat domains with the fork one nucleus domain like the ZFN1. So this is the restriction in the nucleus, and but this domain actually specifies binding with specific DNA, and this domain make a cut. So here also I told you that fork 1 cannot make a cut until unless it forms a dimer. So here you can see left talon fused with fork 1, right talon fused with fork 1. When they specifically bind to your target DNA, then this dimer forms and then it makes a cut. Now talon, you need to engineer a lot for specifically going for gene A, gene B. You have to make different kind of protein. One single tail repeat is nothing but 33 to 35 amino acid. Here is a tail, lot of protein engineering is actually involved to make this talon. So it is also very difficult from repurposing one talent to another talent. So it's also hard protein engineering task. Now comes the simpler one, that is CRISPR-Cas9. And this CRISPR-Cas9 is actually came from a bacterial immune system. And it stands for clustered regularly interspaced short palindromic repeats. Why it is so? So, you know, if you consider this yellow box as a bacterial cell, and we know that bacteria obviously is pathogenic to human and pathogenic to plant. It causes many diseases, as I just told you that Xanthomonas oryzae causes bacterial blight of rice. But bacteria has also enemy. It is also infected by certain pathogen, and for bacteria the pathogen is a virus. Here is an example of bacteriophage, which actually infect the bacterial cell and it destroys the bacterial cell. Now this is an example from Streptococcus pyogenes, and this Streptococcus pyogenes is a bacteria which has an inherent immune system against this viral infection. So how do they operate this kind of immune system? If you see here, whenever there is a viral infection, any virus, they insert DNA into the host cell. This is their normal nature. Okay, if you see our coronavirus infecting human cell, they also similarly they infect their genetic material into the host cell. Here is also this virus insert its DNA or any genetic material into the host cell. Now this host cell has, here is bacteria. Now this bacteria is very intelligent. It keeps a snippet of these viral DNA, and it keeps that snippet of viral DNA as its future memory, because it will know from that sequence that this virus has already infected me. So here you can see this red box is a memory, but where they keep that memory DNA, they keep it in between two repeat. These diamond, black diamond shape are the repeats, and in between this repeat is the memory DNA. Now next to this memory, this is one memory. Suppose next time it has been infected with a virus that is a violet virus, then it keeps the violet DNA here, like orange DNA here, and then green DNA here, and all the memory DNA are flanked with the repeat. And now this locus is called CRISPR locus, clustered regularly interspaced short palindromic repeats. So now this repeat is actually, this locus is a memory locus for that bacteria. So they keep that memory. Now scientists have analyzed that just beside this CRISPR locus there is another important genes are residing. This is called Cas. It produces some that is called Cas9. Here is an example is Cas9, but this Cas gene is nothing. This Cas protein actually is nothing but a DNA chopping protein. It can chop DNA, so it is a nucleus, so it can cut DNA. So now why they are sitting just beside the CRISPR locus? So then scientists have discovered that this CRISPR locus, when it is transcribed, they form CRISPR RNA. These are the CRISPR RNA. Now for each memory, for red memory there is one CRISPR RNA, for green memory another CRISPR RNA, like that, orange CRISPR RNA, then violet CRISPR RNA. Now this Cas9 is a DNA chopping protein, I told you. Then the Cas9 goes and it binds with the CRISPR RNA to form a complex, protein and RNA complex, and this complex is called CRISPR-Cas9 complex, and that's why the name comes from CRISPR-Cas9. So now these CRISPR-Cas9 complex, they remain vigilant inside the cell for any future infection. Suppose now these CRISPR-Cas9, they're all very active, and they're looking for any infection. So suppose now the green virus infects now, and then the DNA, the green DNA comes inside the host cell as usual, and then these green CRISPR-Cas9 complex go and they find similarities. If they find any similar sequences with the CRISPR RNA, then it will bind, and whenever there is a similarities with the CRISPR RNA to the coming DNA from the virus, then the Cas9 gets the signal to make a cut. So whenever there is a homology between the CRISPR RNA and the incoming viral DNA, then the virus actually understands that this DNA is from my enemy virus, because I have that memory with me in the form of CRISPR RNA. So whenever it finds similarities, then the Cas9 makes a cut in the DNA. So the Cas9 makes a cut of the viral DNA. So the viral DNA would no longer be able to damage into the bacterial cell. So this is the CRISPR-Cas9 immune system in a nutshell. Now, if you see in detail of this slide, you would be understanding. So this is the CRISPR RNA. Suppose here green CRISPR RNA, and it binds with the similar DNA. And this DNA is the suppose this is the viral DNA. Okay. So now when the CRISPR RNA binds with this DNA by finding exact similarities with the CRISPR RNA, then it can make a double strand break. Here one break, here one break. But please keep in mind here that it cannot make a cut, or it cannot bind to a specific DNA until and unless the similar sequence has a following PAM sequence. What is called PAM? PAM is a protospacer adjacent motif sequence. For these Cas9 that I am talking about, Streptococcus pyogenes, that is SP-Cas9, the PAM is NGG sequence. This is a 3-nucleotide, and this 3-nucleotide is called PAM sequence. So we have to have an NGG PAM just following the target sequence. Target sequence means the similar sequence. Okay. So in bacterial system, not only the CRISPR RNA, there is another RNA that is called trans-CR-RNA or trans-activator RNA. That is transcribed independently from bacterial own genome, and then this trans-CR-RNA binds with the CRISPR RNA to form a hybrid. So this is a hybrid RNA. Then this hybrid RNA finds similarity and then make a cut. Okay. So now, what scientists have done to repurpose it for our own genome editing purpose. You see, when scientists found that the binding is between only a small portion of the CRISPR RNA with the DNA, so they have emphasized that if we change this sequence to our sequence of interest, so we could make a cut to our sequence of interest. So we could repurpose this system to make a cut to a specific DNA sequence. How? You would be understood from this next slide. So now what scientists have done, this is the CRISPR RNA and this is the trans-CR RNA. Now they have linked the two different RNA by a linker loop. So this is an artificial loop, and then they make it a whole single piece of RNA, and this is called single guide RNA. Now, I told you that this portion is important for binding with a specific DNA. Okay. So now, if you change this one, suppose this one is for gene A. Now you want to target to gene B. Now you have to change this only 20 base pair sequence. So, and the other portion will remain the same. So in normal technological term, we call this one as a protospacer. And this one is a scaffold. This is total about 97 base pair. So you have to change 20 base pair only to target a new gene. Okay. Now, what are the steps to make a double strand break with this Cas9? So first is it will search for a NGG PAM sequence. If it has NGG PAM, then the SP-Cas9 complexed with the single guide RNA, it will bind to that sequence. Now the next step is this Cas9 has a helicase function also. So it will make the double-stranded DNA into a single-stranded loop. So it will separate the double-stranded DNA locally to make a loop, to make a single-stranded DNA. Now, next, the guide RNA, that is called single guide RNA, it will find the homology with the single-stranded DNA newly formed. If it finds the homologous sequence, it will bind with the single-stranded target DNA, and then the Cas9 gets the signal, oh, this is the target that I have to make a cut, because the sgRNA has the perfect similarity with the DNA. Okay, then the Cas9 makes a cut. Now, this was I know this was technically heavy what I described, but now from this slide you would be understanding in a very layman's language. I try to be as simplified as possible. So how it could targetedly inactivate a functional unit of DNA. If you see the CRISPR-Cas9 system, it acts as a scissor, and it has two components. One is customizable RNA, and that is called single guide RNA that I told you, and then another is a DNA cutting protein, that is Cas9. So if you just think about these two components, this Cas9 is like a postman which has two scissors in his pocket, and then this RNA is the address for this postman. So now the postman takes the address and it forms the complex, okay, takes the address where to reach, okay, then it goes and it searches the whole genome to find the similar sequence with the address. So this is the genomic address, you know. Whenever it finds exact similarities, then it will bind there, and then it will make a double strand break in that DNA. So then wherever there is a cut, then the gene would no longer be functional. So the gene would be non-functional. So you are able to deactivate, or you are able to inactivate a functional gene or a functional portion of DNA. So this is as simple as. Now how it helps in creating mutation, or how it helps in genome editing. So if you see, whenever there is a double strand break in the genome, then you know DNA is very important for any cell, and it is a genetic material, so any damage in the DNA is taken very seriously by cell. So now it will immediately, the cell immediately activates its repair mechanism to repair the double strand break. So if you see in normal higher eukaryotes, there are two dominant pathways to repair any double strand break or double strand damage in the DNA. There is one mechanism is called non-homologous end joining, another is HDR, homology directed repair. So this NHEJ is predominant one, and this is the most followed one by the cellular activities. And this NHEJ is also error-prone system. So whenever there is a cut, it always try to repair it completely error-free, but repeated binding, repeated cut by Cas9 makes it erroneous. And as an error, it inserts some random bases or deletes some random bases. And due to these extra base added or extra later deleted from the DNA sequence, then this DNA whole reading frame is getting disrupted. And this disrupted reading frame, the original gene that is supposed to produce a protein like red protein, okay, so now because of this disruption in the sequence, this gene would no longer be able to produce the same protein. So now the function of that gene is gone because of this random insertion and deletion. And that is how this double strand break actually causes this kind of mutation formation. But that is a random, and that happens with very high efficiency. And another pathway is less followed pathway by cellular mechanisms. One is homology directed repair, and that is an error-free repair, but that is very extremely low in efficiency when you try to do that. But that needs
a DNA cutting protein called Cas9 and an RNA molecule known as the guide RNA. Bound together, they form a complex that can identify and cut specific sections of DNA. First, Cas9 has to locate and bind to a common sequence in the genome called a PAM. Once the PAM is bound, the guide RNA unwinds part of the double helix. The RNA strand is designed to match and bind a particular sequence in the DNA. Once it's found the correct sequence, Cas9 can cut the DNA. Its two nuclease domains each make a nick, leading to a double-strand break. Although the cell will try to repair this break, the fixing process is error-prone and often inadvertently introduces mutations that disable the gene. This makes CRISPR a great tool for knocking out specific genes.
Okay, so I think it is a bit clear to you now. So what we need to do actually to do a practical genome editing using Cas9. So you have to insert some DNA sequences into the cell. Here I'm giving an example of DNA sequences like plasmid DNA, but I will be giving some other examples also. So here you can see this is the Cas9 forming, Cas9 protein forming sequences. This is Cas9 DNA, and this is a promoter to express the Cas9. And another component is that this is the portion that will produce the single guide RNA. So you have these two components. When you insert these kind of plasmid DNA into your cell, so this portion will produce the guide RNA, and this portion will produce the Cas9 protein, and it will do the job for you. Okay. So now, if you see a real example from a real experiment, so suppose this is a target 20 base pair sequence that you want to make a cut. So here you can see this is the target. The red one is target, and it is followed by the 3-letter PAM. N means any nucleotide. Okay, either A, T, C, G. So N is C, and then GG. So NGG is the PAM for Cas9. So this PAM is present here, so it is targetable by you, targetable by the Cas9. Okay, so now you can target this sequence. So you can make a cut. Pink means T, and this black means G, blue means C, and the green means A. So you can see this is a wild type sequence that is not treated with Cas9. This is a normal one, not mutant one. Now when you treat this plant with a Cas9, then it produces this kind of sequences, and you'd get to know that Cas9 made a cut, and then it made an error, and you got a mutant plant with single base insertion. How is it? If you see the sequence here, so here the A was not there in the wild type sequence. You can see AACC, ACGG. So the A is inserted here. If you compare these two chromatogram, you can see peak here means 3C, but here within this 3 blue peak there is another green peak inserted. It means one nucleotide inserted erroneously, and that insertion causes the disruption of whole open reading frame of that gene. So that's why this gene is mutated. So this gene will not produce the same protein as it should produce in the normal condition. Okay, now if you ask me, okay sir, this is very easy to do one single mutation in one targeted gene. So how about doing more than one gene in one experiment? So this is called multiplexing. So in case of Cas9, the multiplexing is very easy, because you need to design same kind of plasmid vector with the same Cas9, but you have to give 3 addresses to the postman. So the Cas9 takes address for locus 1, and then Cas9 takes 3 address separately, and then they go to locus 1 genome. So it would disrupt this gene, and this gene. So like that you could achieve at least up to in our lab in USA we achieved 8 different simultaneously 8 different gene mutation in a single experiment by giving 8 different guide RNA. So 8 different address we have to design, so it would mutate all 8 genes in one go. This is called multiplexing with Cas9. This is very easy. Now let me just show you some limitations of this technology. So it has some problem with off target, obviously. In many cases, in most of the cases is very insignificant, but there is some off target editing possibilities. So this is one limitations. Off target means suppose you targeted gene A to cut, but it erroneously, suppose in 95% cases or 99% cases it made a cut at gene A, but in 1% cases, and less than 1% cases it could made a cut to other gene also. That is called off target editing. So So now the next limitation is you have to have an PAM sequence just followed to your target sequence. So this NGG should be there always in order to make it a targetable sequence. So this is another limitation of this technology. I would tell you how to avoid these conditions. And then another limitation is you know, this Cas9 is pretty big protein. It has 1368 amino acid, and if you see the nucleotide length, it has 4100 base pair, so 4.1 kb. And in normal human genome editing is done by this adeno-associated virus, or AAV virus, and which has a packing limit. Packing limit means it has a limit to contain DNA inside it. So actually, this is a vector. This virus is, you know, I told you that virus always insert its genome into the host cell. So suppose you keep Cas9 and sgRNA, DNA inside this virus, and this virus goes into the human, and then it inserts the Cas9 human cell. Okay, this is simple. So, but it has a limitation because it has a packing limit of 4.7 kb, but I told you Cas9 is itself 4.1 kb, and then you have a promoter, and then you have guide RNA sequence and all the other stuffs. So it is very difficult to pack the whole stuff into a AAV vector. That's a limitation. Another is it has a low efficiency of precise editing. So how obviously Cas9 is very precise because you want to target to gene A, it goes to gene A because it has the same similarity as the guide RNA. But what is precise editing? I am talking about precise editing means suppose you don't need to knock out a gene, but you want to change specifically certain base pair to another base pair. Suppose you want an AT base pair into a G editing. Until a few years ago, it was very difficult with Cas9, but I will talk about later more. So if you see about the first limitation, I told you that off-target problem. So there are a lot of, you have to design your experiment very, very cautiously because there are a lot of software tool available. So this is a snapshot from one of the software that's called CRISPR-P. So these are the, suppose you put a gene sequence, you know that gene sequence, you could download it from NCBI, and that gene sequence you put in that software, and then it will design several probable guides here. These are the all 20 base pair guides with the PAM sequence. Okay, they're on score. This on score means their cutting efficiency, predicted cutting efficiency. Now, if you click in one guide, you will get in the right hand side off score. So you get this at the off score for this guide. So now you have to select among this whole lot of guide which has the least amount of off score. So then you have to go for that off score. Less off score containing guide. Okay, so this is one way of avoiding the off-target editing. But obviously, after you successfully edit your suppose gene A, you have to also go for analyzing what are the other off-target sites, whether they are edited or not, because you can predict the probable off-target. You can see how this off-target arises. You know, if you have a 20 base pair, the 5' end of the single guide RNA, this 20 base pair, if I told you that it should be all perfect homology to make a cut, but there are some error if there are suppose 18 base pair homologous, but 2 base pair is non-homologous. In that kind of things, sometimes Cas9 may also erroneously cut that wrong sequence. Okay, so that's why off-target arises. Now the next limitation was I was talking about NGG-PAM. You should have an NGG-PAM. Now these technology is going so fast that every day there are a lot of papers is being published. So there are a lot of effort to engineer the SP-Cas9 to make it PAM. So if you see, NGG-PAM is not required for this kind of 3 engineered Cas9, where only a presence of NG is sufficient to make a cut. Then this is Streptococcus aureus Cas9. It has a different PAM requirement. This Cas12a has TTTV PAM. So if you have a different PAM like TTTV, then you can use Cas12a like all other things. So we have enlisted in our upcoming chapter, one chapter we have written for CRISPR-Cas method Springer book, and we have enlisted all different kind of orthologous Cas9 means and then also engineered Cas9 that has a different PAM requirement, not the only NGG. So you have a lot of flexibility to use from the whole CRISPR toolbox. Now, the another problem, I told you that delivery is a problem of this Cas9 for human genome editing. So there are several methods that scientists have tried. One is to inserting into the genome, into the human cell, the whole mRNA that will produce Cas9 when it will go inside the cell. So it will be the human cell would be transformed with mRNA and the guide RNA, and when it goes inside, that mRNA will produce the Cas9 protein, and it will take the guide RNA to form a complex, and it will go and search for the similarities and make a cut. Similarly, in the second way, here you can see RNP could be also used. RNP is ribonucleoprotein. Ribonucleoprotein is a combination of protein and RNA. So Cas9, purified Cas9 protein you have to take, and then you have to the guide RNA. So guide RNA and the protein, you complex it outside, and then you insert into the human cell to make an edit. And then another is very intelligent engineering, is that you can split the whole Cas9 into two pieces. So the coding DNA is split into two pieces, and then one piece it inserted into one AAV vector, this virus, and another piece in another virus, and then you co-infect with both kind of virus into the human cell, and then when it goes into the human cell, then the protein would be formed separately, and then the protein will come together to form the whole Cas9, and then when the whole Cas9 is formed, then it will go to the target site. So this is called intane split Cas9. This is also very interesting. Okay, so what is low efficiency of precise editing? I was telling about this precise editing. Precise editing means it's not only knocking out or inactivating a gene. You have to correct a gene sequence. Suppose you have a wrong sequence, you have to make it a correct one. You don't have to inactivate it. So this kind of precise editing is mainly done with HDR, but the HDR is very low in efficiency. But there are two technologies recently discovered. One is base editing, another is prime editing. I will talk about details about base editing because I have mainly worked on base editing. But we just started working on prime editing in our institute now. But I am not talking about prime editing today because time is limited. So now in part two, now coming to part two, what else we can do with CRISPR-Cas system beyond knocking out a gene, beyond inactivating a gene. So you see, I told you this postman or the normal Cas9 has two scissors and it can make double strand break. But if you change this Cas9 a little bit, sequence of this Cas9, you can make it an engineered Cas9 that is called Nick's Cas9. So this Nick's Cas9 has only one scissor, not two scissors. So it can make only one single cut, not double strand cut, and this is called Nick. So this is called N-Cas9 or Nick's Cas9. Now you can make this Cas9 dead. How it will make, you will make it as a dead because you just take out both the scissor from that. So how to do that? This is a protein engineering. So two single mutation in the Cas9 DNA would make the Cas9 ineffective to cut because it would not have the ability to make any cut, but it would have the ability to bind to certain sequence because it has the ability to bind to the sequence but not making any cut. Now, if you want to play with gene regulation, we need to have those kind of N-Cas9 and D-Cas9. Suppose this is in order to express a gene, in order to produce protein from a gene, you have to have this kind of cassette where promoter actually controls the expression of gene and terminator actually stop the expression of that gene. So if you see, this is the expression cassette. So now this is a D-Cas9 or deactivated Cas9. So when this D-Cas9, if you give that D-Cas9 the RNA or guide RNA, that is the address for that D-Cas9. So this D-Cas9 will go to that address and it will bind, but it will not make any cut because this is a deactivated Cas9. Now if you fuse the D-Cas9 with another protein that is called CRAB, and CRAB is a very known inhibitor of transcription. We know transcription produces RNA from the gene, okay? So if you give these D-Cas9 fused with CRAB, RNA address, that's address actually directed to the promoter of a gene. So this dCas9 would go to the promoter or the enhancer and it will bind there, and because of the presence of this CRAB that inhibits the transcription, so the gene expression would be inhibited, and this is called CRISPR interference or CRISPR. Now another version of this kind of technology is called CRISPR-A or CRISPR activator. So the same technology, you fuse the dCas9 with BP64. This is viral protein 64, is a known activator of transcription. So if you fuse dCas9 with BP64 and give the address means the RNA, then it will make a complex and go to the promoter, and it will activate the expression of that gene. So this is called CRISPR activator. So you could reduce the expression of a gene, you could activate the expression of a gene. So now another form of activating a gene is, so you know, this is called epigenome editing. So in many cases, some genes in one species is very down regulated, so it is not producing that much of RNA or protein, but the same gene is present in another species, but it is producing a huge amount of protein or RNA. But why this kind of difference? Because the same protein has same promoter. The promoter region is methylated. The bases in the promoter region is methylated. So the methylated bases actually inhibits the expression of that gene. So this is called epigenome signature. So if it is methylated, it is down regulated, so it does not produce that much of protein or mRNA. But if you have this kind of problem, CRISPR has that answer also. You have to fuse the dCas9 with a TATE1. TATE1 is a protein that could demethylate. So when you fuse TATE1 and with the dCas9, and if you give the address to the promoter of your gene of interest, you know that this gene promoter is methylated. So you target the dCas9 fused with TATE1 to that promoter, and it will demethylate. This blank round shaped structure is signifying the demethylation. So the TATE1 will perform the demethylation, and what dCas9 does here, dCas9 actually takes the TATE1 to that promoter. Okay, so this is called epigenome editing. So like that, you could also increase the gene expression of a gene by editing the epigenome portion. Now this is another very interesting nowadays, because we all talk about coronavirus infection, their detection, their diagnosis. So you know, this kind of virus, when we are infected with a very low copy number of virus in initial stage of infection, it is very tough to detect whether this virus is present or not, because in 1 microliter of blood, maybe one virus copy is present at that time. So it is very difficult to detect. So this small numbers pose a large problem for medical diagnostic. Now CRISPR came for this kind of problem. So there are two methods. One is called detector method, and another is called Sherlock method. They devised, one is from Jennifer Doudna from UC Berkeley, another is from Feng Zhang from MIT Harvard, and both are depend on two orthologous Cas protein. That is, one is Cas12a, another is Cas13. So this Cas12a has some special properties, not like Cas9, too much specific. Obviously, this Cas12a will go and bind to the target sequence if you give a guide RNA. Okay, but it has a collateral activity. Collateral activity means it will bind to the target, it will make a cut, but it will, when it finds the target and successful binding, makes the Cas12a also cut other adjacent DNA if they are present. So if there is another DNA present in the media, it will also make a cut. So you see, if you give this kind of DNA in the tube, suppose this one, with a coencer, with a reporter and coencer, okay, so this is a reporter dye that will fluoresce, but in presence of coencer means another suppressing thing. So if it suppresses the presence of these dye, when they both present, they will not fluoresce. But when only this one is present, then they will fluoresce. So suppose your tube has a viral component, you don't know that, you add Cas12a virus component, or suppose blood serum, and then you add this reporter coencer dye. So now and you design the guide RNA to target the viral DNA. So now, or viral RNA. So now when this viral DNA or any DNA is detected, especially homologous to the guide RNA, then this Cas12a make a cut in the target also, and also the coenchain reporter one. And this reporter becomes free, and then it will fluoresce, and it will get a detectable method to ascertain that this kind of DNA sequence is present there. So this is used in CRISPR detection. So you can see there are at least 4 different very good quality of application. So this is CRISPR-Cas12 based detection of SARS-CoV-2 published in Nature Biotech, and this is another Sherlock method based on Cas13 from Fang Zhang's lab. This is named as Stop COVID. So these are 2 detection methods. I'm not going into details of this one. So these are a whole lot of things here, you know. Like you can do DNA live imaging with CRISPR fused with GFP, and then you can change the chromatin topology, and then you can target also RNA targeting, and then CRISPR screening. So you can screen thousands of genes in one go with the CRISPR, and then you can also reorganize the genome, and you can also use CRISPR associated transposes and then gene drive to actually eradicate a whole population. And this is very important for getting rid of those malaria kind of causing mosquitoes. So you know, this is also has been devised with CRISPR, this gene drive technology, and World Health Organization is also thinking very seriously to use gene drive to eradicate those malaria causing mosquitoes in the African countries. And then also rapid domestication in case of plant, and then prime editing. But I don't have time to explain all those things, but these are the things just to enlighten you with how much we could do with this technology. But this is not all. There are a lot of different things. So now this part 3, I would just briefly go through my experience with precise editing. So this is a simple movie clip from an Indian movie that's called Pa, and this is Amitabh Bachchan, you know, and he was 12 years old, and his friend was also 12 years old, and you see the difference. And this is called and his name was Oro, and Oro was suffering from progeria disease. Progeria is a rare genetic disease, and that causes rapid aging in the childhood. So that child look like a very aged and old person, you know, and that is because of what, you know, why he is so different, because there is only single nucleotide difference in a special gene, and the Oro has in a certain position a nucleotide, whereas the healthy one has a G nucleotide. And because of this difference, Oro has died at 13. And Oro is not alone. There are a lot of child that are affected with this kind of rare genetic diseases. Okay. Also, this is another example for plant. If you see, this is a Kitake rice genotype, and this is a mutant. So because of the single difference in a gene, it has a C nucleotide, whereas this one has a T. Because of this difference, you know, this is less affected with the disease, whereas this is affected more. So this kind of single difference, single nucleotide difference could make a huge difference in the expression and make a lot of human diseases and also plant diseases. So then our question is, long-standing question is there a way to rewrite genetic letters? You know, we know the reason of Oro's suffering because of these changes in his DNA, but is there a way to rewrite genetic letters? The answer is yes, because of this discovery of base editor. So this adenine base editor causes or could change A to G. If you have an A, you could change it to a G by using adenine base editor. And there's another base editor is cytosine base editor, which could change a C letter to a T letter. So this adenine base editor here, I already introduced you to N-Cas9, Cas9, okay? This Cas9 is fused with this adenine deaminase. This is laboratory evolved deaminase protein. It is fused with the N-Cas9 to make this kind of changes in the DNA. I'm not going into detail of the mechanisms, how it causes. Maybe next time, if I have time, I could explain how it actually changes the DNA, single nucleotide. Now, for cytosine base editor, similarly, the N-Cas9 is fused with cytidine deaminase. This is R-ApoBSC1. This is a rat ApoBSC1, it's rat cytidine deaminase fused with the N-Cas9, and it could change a C to T. Now, this is David Liu, who has actually pioneered in inventing this technology, and I was able to meet him. Okay. So you'd be understanding from this video a little bit more.
Then be used to transport those enzymes to a specific DNA sequence. In one example, Cas9 is fused to an enzyme, a deaminase, which mutates specific DNA bases, eventually replacing cytidine with thymidine. This kind of precise gene editing means you could turn a disease-causing mutation into a healthy version of the gene.
Okay. So, as I told you, the precise editing, if you could do it before the base editing invention, you have to go for homology-directed repair. So, you make a cut with the Cas9, and then you supply a donor template with the change. What change you want to do? Suppose you want to make a change from A to G, like the case of Oro's case. Okay, so A to G. So you make these changes here, and then you supply the donor template, Cas9 and guide RNA. Then you get the precise editing, but this is extremely low in efficiency. So if you see, in case of animal, there are reports that it ranges from 0 to 5% editing. So this is very, very low in efficiency. But in plant system, it is much lesser, because plant cell has a cell wall barrier, so it is very difficult to supply this donor template in adequate amount for this kind of precise editing. So but base editing actually solved this problem of precise editing. So what this is adenine base editor, it could make, I told you that A to G in one strand, in the opposite strand, if you see, it could make T to C changes. Okay, so I have worked with this adenine base editor, and we selected two different genes. One gene is white stripe leaf 5, it is in rice, and this single mutation, this T to C mutation in this gene sequence causes the leaf becomes white striped. Okay, this is a single letter changes. Another change is another gene was Zebra3, and it is a mutant also because of a single letter change in the gene sequence, that is also T2C. So if you have this kind of change, a single letter change, then you get this kind of phenotype, dark green, light green, dark green, light green, dark green, light green, this kind of tree. So it's called Zebra3 mutation. So we targeted both of these loci to edit with the ABE. So I just want to go through very quickly. So we multiplex this experiment with an advanced system developed in our lab, that is called PTG, polycistronic tRNA and guide RNA. So this is a guide RNA. This is the 20 base pair for WSL5, and this is the scaffold, and then another tRNA, and this is the 20 base pair for Z3, and this is the same scaffold. So this is 20 base pair for WSL5, this is the 20 base pair for Z3, and now when it is transcribed, so the cell has any eukaryotic cell has these two enzyme, RNase-P and RNase-Z. So these two, they cut out the tRNA, and then when this tRNA is cut out, then this guide is separated from the other guide. So then this guide will make a complex with the base editor, and it will go to the chromosome 4, because the WSL5 is present in chromosome 4, and the other Z3 guide, it will also make a complex with Cas9, means N-Cas9, that base editor, and it will go to chromosome 3, because the Zebra3 gene is present in chromosome 3. So you could multiplex in a single experiment. So we have done it too for this base editing, but you could do it more. So now, if you would be amazed to see this result when we compared the chromatogram sequence with the control one, with the mutant one. You see, this is the control one. So we targeted this A to change into a G, and we treated with the base editor, adenine base editor, and you can see all the other peaks are similar as like the untreated one, but this one is specifically precisely changed. This green peak has been changed into a G means black peak. So the green peak changed to a black peak, that designate A has been changed precisely into a G. And because of this targeted change that we wanted to change, we got this kind of expected phenotype, the white stripe leaf. So you see, you can imagine that this technology is able to change specifically the single letter of targeted single letter of your gene, and it could translate into a phenotype. Now for the next example, similarly the Z3, also here our target was this A and we wanted to change it into a G, and similar result we have found that green pig has been changed to a black pig. And because of this same precisely change, you can just also compare the all other pigs are not changed, only this one is changed. So this is also targeted single letter change in the gene. So because of this base editing, we also got similar kind of dark green, light green, dark green, light green phenotype here, and the mutant phenotype, other mutant phenotypes also observed. So we could achieve successfully two different loci edited precisely single letter at each of the gene. So now in a very short time we could develop a transgene-free mutant obtained in a single generation. How? Because someone has asked in one of the audience has asked how it is GMO free, it is not transgenic, it is. So this slide would answer this question. So you know, suppose you introduced your base editor construct and this base editor is integrated in chromosome 1. Now your target is in chromosome 4. So now this chromosome 1, here is the DNA for base protein. So now this DNA would produce the base editing protein, and this protein takes with the guide RNA and it will go to the chromosome 4 to change the single letter. Okay, now you see your target was here, so when it is changed you don't need base editor to be present in your cell. So in the next generation, in sexually propagating cell, if you grow the seeds, so some seeds will contain it is called segregation, it's called Mendelian segregation. You know, the base editor would be segregated out from the targeted mutant. So in a few generations you would get only the mutant plant but not having any kind of inserted DNA. So base editor, you don't select for base editor, you select for only mutation. So if you see, this is a simple example. The absence of this band means absence of base editor. So the black band means presence of base editor. So out of this 25 plants, this does not have any base editor, but these all 6 plants have the targeted letter changed. So they are mutant, but they don't have that base editor exist. So these 6 plants are non-transgenic, they are non-GMO. Okay, so this is how you could obtain a Cas9 mediated non-GMO but gene edited mutant plant. Now with this base editing you could do a lot of things. If you apply CBE, that is cytosine base editor, you could make artificial stop codon. So you could make stop codon. Now I gave you an example with adenine base editor how to change in a stop codon to a coding codon. So if you have this kind of stop codons, then you can change them to a coding codons. So you can correct or create stop codons with the base editing technology. Now you know, in normal eukaryotic splicing there is 2 important things. One is a splice donor and splice acceptor in the intron. So this is called GT. This GT is a donor sequence and AG is an acceptor sequence. So if you apply base editor, BE signifies base editor, and if you change this sequence, then you would play with the splicing mechanism and it would give rise to different kind of proteins. And this is exon skipping, alternative splicing, and intron retention you could achieve. Now this has huge application in disease modeling and therapeutics. How? Because you know, in human disease we don't always analyze an infected human being to better study that disease. We create a model, disease model that mimics the same kind of genetic sequences like the human. So these are the different models, but it was very difficult to make certain disease model like progeria disease model. It needs a single base pair changes. So how would you artificially make a model? Because we did not have any technology to make that kind of changes. But because of the base editor invention, now it is possible to make that kind of disease model in mouse, in rabbit, and fish, and nematode, all these things. So you have a disease model. Now you could better investigate about the disease, and then you could devise a therapeutic strategy. So this is about modeling. Now base editing is very unique because this could be applied starting from the embryo, blastocyst, and fetus, newborn baby fetus, and then even non-dividing cell like an adult. In adult human being also, this base editing could be applied. So now, you would be amazed to know that almost 61% of pathogenic human mutation could be corrected with this base editing technology, and it has already been demonstrated that those are all different genetic diseases that has already been cured in human cell line or in disease model. So now we have an answer to cure this kind of child from suffering from progeria. So now this is a very exciting example recently came out in this 3rd June 2020, that base editing restores hearing in mouse model. So so this is a deafness, you know. So this deafness is caused because of one mutation. Now base editor has been applied to restore that single base mutation, and you can see this is a normal cochlea hair cells that is very essential to have a good hearing ability. But you can see this is the deaf cochlea hair cells from the deaf mouse, but when it is treated with the base editors, it has already been almost corrected. So, and it got its hearing back. So this is a very exciting news. This came out few days ago with the base editing. So it has already been applied in various agricultural crops for various different kind of applications that we could do. And now this is another kind of precise editing by predicting what kind of mutation it would generate, that CRISPR-Cas9. I told you that CRISPR-Cas9 normally due to during knockout mutation, it produces some kind of random insertion and deletion, and that's why it is mutated. But by using machine learning mechanism, by using machine learning algorithm and all these things, if you have a big repair data and if you feed in model training, then this kind of model predictor models develop, and then you could select the guide RNA which it produce, and which could also, this model also predict what kind of repair it would produce. So which guide would be better, and what guide would give rise to what kind of editing. So to give you a simple example, suppose then this is the PAM sequence and this is the 20 base pair target sequence. If you have at the 4th position a T, then this guide would produce more likely another T would be inserted. So this is a single base pair insertion would happen if the 4th base of your guide has a T, like that here. If you have a double C at -4 and -5 position, then it is likely that you would lose one C, so one C would be deleted. So this kind of prediction could be done with this machine learning. So we have wrote a forum article for this, Trends in Biotech, so you could go through that. I would give you reference later. So okay, so in order to break the monotonicity from my lecture, this is a very exciting video. I want to see a real-time atomic force microscopy video, how Cas9 is cutting in real time the DNA. So here, so this violet, this is the Cas9, and this is the DNA thread it is bind to it. And now you can see it is cut. It is cut after a few times. This is the Cas9 bind with one portion of DNA, and this DNA is cut already. So this is how Cas9 works. And now I'll give you a few quick outstanding examples. So this is Waxicorn developed by this is a starch biosynthetic procedure. So here you can see from ADP glucose, it goes either amylose or amylopectin. So if you inhibit the amylose production, it would produce more amylopectin. So they have deleted using Cas9 these amylose producing gene WX1, and then they produce more than 97% amylopectin containing corn, and it is being commercialized in USA, and it is very highly valuable for processed food adhesive and high gloss papers. So now okay, so this is a non-browning mushroom. You know, if you cut a mushroom and if you keep it for longer time, it becomes brown. Okay, so this browning happens because of the PPO genes, that is polyphenol oxidase, and if you reduce the expression of that gene, then it would no longer be brown as soon as the wild one. So this is my supervisor that I worked with in Pennsylvania State University, Inong Yang. So he did in mushroom, and he produced non-browning mushroom by knocking out 6 PPO genes in this agaricus species. So this is another example of soybean with high oleic acid, 80% oleic acid, which is very good, and then 20% less saturated fatty acid. You know, this is both healthy, and also storability is high than the normal soybean oil. But this has been used this has been actually developed by utilizing talent technology by Dan Porteous group from Minnesota. So this has already been commercialized, and it is available in USA market. So you could buy this genome edited soybean oil that is called Calino. Okay, so now this is, you know, hybrid rice seeds production. So normal hybrid rice could not be saved by farmer, because each year they have to buy new seed for getting that hybrid production, with very high production. But this technology, CRISPR, has already been used by 2 different labs. One is from USA, another from China, and they demonstrated that using CRISPR we could produce clonally, so we don't have to buy each year from the company. We can save the hybrid seed and we can grow in the next season. So this is a unique and very, very useful commercial application of CRISPR-Cas9. So now it is to make a plant resistant to DNA virus. So here, suppose you transform your plant cell with Cas9 and give the guide RNA or the address. That address is similar to the viral DNA, suppose. Then suppose your cell has this Cas9 and the guide RNA which target the viral DNA. So whenever the virus infects, then these Cas9 and guide RNA go and cut the viral DNA. So your plant would be resistant to that virus. Now the next question, you may ask me that, what about RNA viruses? If the virus is RNA, what would happen? Then the Cas13 is a brother of Cas9, is an orthologous protein of Cas9 that has the same property of specificity, but that do not bind to DNA, but that bind to RNA and that chop out the RNA. It's like Cas9, but it is for RNA. So you can use Cas13 to target an RNA virus. Okay, so this is a very good example of developing bacterial blight resistant rice. So you know, there are some genes which is called susceptibility genes that help pathogen to establish themselves, and it modulates the host defenses and pathogen sustenance. So it is present within the plant, but it helps the pathogen. This is called susceptibility gene. So here, in case of Xanthomonas, they secrete this tail protein. I told you earlier, this tail protein goes into the promoter and it activates this kind of susceptibility gene, and it expresses in higher amount. Whenever this gene expresses in higher amount, it actually facilitates the Xanthomonas, because this gene is a sugar transporter gene. For sugar to the apoplast, so the bacteria will have more food, very intelligently using plant's own gene to better survive into the plant cell environment. Okay, so now we know this mechanism very well. So it has been used, TALEN also and CRISPR also, to mutate this promoter sequence where the tail binds, where the effector binds. So if it is mutated, then this effector could no longer bind with the promoter, so the sweet gene would not be activated, and that has been recently published in 2019. So these are the result. Now, what about therapeutics? These are the 3 things that are under CRISPR clinical trials. There are a lot of examples of different disease correction in disease model and human cell lines, but they are not in clinical trials. But these 3 are in clinical trials. One is making cancer killing T cells, that is highly active T cells. And another is 2 blood disorder like sickle cell anemia and beta thalassemia, but those are all ex vivo editing, not in vivo editing. What is ex vivo editing? So you have to isolate the whole the blood from the patient, and then you edit outside the patient body in vitro the blood cell. You edit the blood cell, and then you again insert that blood cell into the human body. So it's kind of, it's safe. It's not it's not that you are inserting files with the cancer killing T cells. The first phase result has already came out, and it has been shown that after 9 months of that treatment with the CRISPR, it is not showing any adverse kind of effect in human. So it is a good sign for CRISPR scientists. Now, to know more about some work, so we read a very good article for Cell Press. It's for Trends in Biotech on the base editing technology especially. So you could go through that. And then we wrote about predicting the CRISPR-Cas9 induced mutations. So what kind of guide would be better, and what kind of mutation it would produce. So that kind of thing we have discussed in this forum article. And then yes, I have already discussed about this 2 gene editing that is already published. You could go through for having a better idea. Those are freely accessible. And then this book is going to be published in the late June, this June. It is in press, already proofread has been done. So there are all methods we have edited this book. How to perform, protocol is given in this book, how to perform different kind of editing. And this is to start with, you could go through my blog. I have wrote for AdGene, CRISPR mediated plant-based editor, and this is written in a very layman language because this is intended for students and those who are new to the field. So this is not a technical one, so you could go through this blog also. So now in conclusion, what I could say for part 1, part 2 and 3, that genome editing has unprecedented ability to generate targeted and specific mutations in almost every loci. Knockout, point mutations, gene regulation, and generation of rapid targeted genetic diversity is possible with this technology. And if you see the examples that I have given, it is easily understandable that these simple, versatile, and efficient tools revolutionizing precision plant breeding, biological studies, and human therapeutics.